Ruey Chih Lee, and I'm the student who attended this class last year. And because the numbers of our class is not enough, so today's presentation will be me and the other student who also attended the class last year to demonstrate how to present the paper. And okay, today my topic is use of machine vision to sort tomato seedling for creating robots. And here is today's outlines. First of all, I will talk about the introduction and then the methods for the grafting robots. And after that, I will show you the results and the discussion. And finally, I will talk about the conclusion of this paper. And first of all, what is grafting? Grafting is a method of asexual plant propagation where the tissues of the plant to be cultivated like this picture, like cyan, and are encouraged to graft onto a strong vital plant like this rootstock, and so that the cyan can grow and blossom well. And also there are some papers found that grafting plants were taller and more vigorous than cell-rooted ones. So there is some advantage of grafting. It's easy to propagate, and it has variety improvement, and also it can increase the production. So here is the main purpose of this research paper is to develop a grading and sorting algorithm for a fully automatic grafting robot. And in this paper, we will grade and sort the tomato seedling into 4 groups using the machine vision for these grafting robots. And then to make seedling to be grafted more precise and faster. And but in the past we graft plants by ourselves by hands, and it is a time-consuming and tedious job, even for skilled workers. So this is the scope of this study to this. And then the methods of this paper. About the grafting operation, we need to select the same diameter of the cyan and rootstock of the seedling so that we can graft well. So we need a camera to substitute the human eyes for grading and sorting the seedling. And also we are in this machine vision system, there are two major categories of artificial lighting source. One is the figure A, is lighting the object from its front side, and which means from the camera side, so that the required images are visualized to the camera. And the figure B is lighting the object from its back side, so that the shadow of the object is visualized to the camera. And in this paper, we have to find a suitable one for this study, so both of type of this lighting source will be tested. And also we need the backlight color, and there are 3 colors, white, blue, and red colors panel backlights. And also we will test all the 3 color backlights were separated used with the camera to acquire the images. And about the light filter, because lighting source produce some reflective or halation effect on the object, so which makes the certain parts of the object highly bright. So we need the light filter to remove these halogen effects, which can make the image more clear. So the polarized light filter are often used while acquiring images. And there are three different combinations of the device. For 10 centimeter camera distance were used to acquire the image. And one is using no filter, and one is using PL filter on the camera lens, and one is using PL filter on the camera lens, as well as on the backlight panel. Okay, and also we are going to compare to find the best combination in extracting the required features of the images. Okay, and here is the setting. This figure is the setting of this image acquisition, and as we say, there is a backlight panel, and in the middle it will be the tomato seedling, and the camera is in here. Grading and sorting the seedling, we will grade the sort the seedling, we will into the 4 groups. And group A which is the diameter of the seedling is more than 2 millimeter, and group B is between 1.6 millimeter to 2 millimeter, and the group C is the stem diameter range is less than 1.6 millimeter, and the group D is substandard, which is like this figure, which means we cannot distinguish the stem and the leaves because there are the irregular arrangement of the leaves. So we cannot this image cannot be analyzed. So it was graded as a D category and was left for manual grafting. Let's show the results and discussion. Here is the results of the illumination source, and about this figure, we can see that along the pixel horizontal axis, the gray values of the pixel were extracted from both the images that were acquired from the front lighting as well as for the back lighting. And the bottom part of these two curves indicate the seedling diameter. And this range will indicate the background of the image. So in this figure, we can, in the backlighting system, we can distinguish the background and the diameter well. So the backlighting system was chosen for acquiring images of seedling. And then about the backlight color. In this figure, we also the blue backline is more sharp than the other two. So the blue backline in this consideration found the best among the three. And about the PL filter, see the without PL filter these curves, this one. This indicates that no PL filter was used with the camera or with the backlight. And the edge of the seedling stem was brighter due to the halation effect of illuminations. So about these two curves, which is also which are used the PL filter, and it were sharp compared to the previous one. Previous one means without the PL filter. So this behavior proves that the halation effect could be successfully reduced using the PL filter. So in this paper, it was designed to use the PL filter with camera lens as well as with backlight panel. Okay, here is this image is the seedling image in the binary scale, and figure B is the counting binary values of the pixel along the horizontal axis. And we can see that around the pixel number 250, which is indicated that the stem diameter and the pixel increase gradually up to the pixel number 360, which indicates the presence of the these small leaves, yeah, this one. And up to the accumulated number indicates the maybe around the 550. And this is not of the true leaves. And yeah, and about this figure, we can make some image processing to gradient sort the seedling. And in this figure, which is we cannot distinguish the leaves and the stem. So in this case, there will be grade as the group D, which is substandard, and will be left for manual grafting. Okay. And then this table shows the algorithm. Could source 97% of the seedling successfully, and the rest 3% was failed because the target is shaped by the irregular arrangement of the leaves, as we saw is figure. So we cannot this target cannot be sorted by this algorithm. Okay. And it also found that most of the sorted seedling had larger stem diameter, which are desirable for proper grafting. So there is the conclusion of this research paper. And an algorithm of grading and sorting tomato seedling using the machine vision is developed for a fully automated grafting robot system. And it is observed that backlighting system is combination with PL filter is better to recognize the shape of the seedling in the images. And also in this paper, the success rate of the algorithm was 97%. And the rest 3% settling was failed because of the irregular arrangement of leaves. So this algorithm is not bounded for tomato seedling only, and it can have the other application maybe used for the paper, eggplants, or so far. So we can more. It can give the potential of the grafting and to replace slower manual grafting. Okay. Here is my presentation, and thank you for your listening.
Thank you for your great presentation. So any questions and comments? And to you? And how I want to ask about the sorting rate. How long will it take to scan one plate? How long will it take to take an image of the plant and analyze it and analyze it? Because the purpose of this paper is to enhance the working rate. So
You mean the whole processing of the system? How long?
right? Just one. Because you need to compare this one and with the made by human hand.
I think the paper did not indicate show us the exact time of the processing of the system. I think because we have to make sure the same diameter of the spine and the rootstock and should be matched. So in the past we use the eye vision, and in this system we use the camera. So I think it should be more faster.
Okay, thank you. More questions? Please,
Do you think, or do you know if it's applied in real life today, or do you think, if not, do you think it will be used in the future, this method? Maybe I should search the more paper of this machine vision, but I think it is benefit to environment.
So, more questions from Kyoto University?
Okay.
Or comments?
Okay. Are there any questions? Okay.
Hello. You mentioned backlight system, and there are three colors: white, blue, and red. And could you tell me which color is most useful for recognizing the shape of seed line, and why the color is most useful? Thank you.
As I talk, these three colors red lines were independently used with the camera to acquire the images, and and the result of the test is shown this figure, and the paper say the weapon of the blue backlight is short. The weapon of the blue backlight is short, and it cannot penetrate the plain tissue. So we there will be the other reason to choose the blue backlight in this paper, I think. Did I answer your questions?
Okay, thank you.
Thank you.
Okay. So, another question from Kyoto University. I'd like to know how you would be able to sort a seedling which has lacked water. Instead of grafting, maybe the seedling has lacked water or minerals. Will you be able to graft it again, or the system is able to recognize that this one needs more water instead of need for grafting? Thank you.
I think it can be analyzed by this system because we are take a picture of the plant, and then to make and the image will be converted to the binary scale. So I think it can be analyzed by the algorithm of the sorting and grading. Yes.
で、今回は、次から。
Thank you. Any other question? Okay.
You said the success rate of algorithm is 97%. I wonder, is it enough high for practical use?
The paper didn't mention the question you asked, but I think it is. I think it is. I think the method of the grading and sorting algorithm is has the potential to yes.
Thank you.